I'd like to thank Sophion Bioscience Science Company and advisory board members for giving me the opportunity to talk about my research today. In my talk, I will first talk about, as an introduction, about regulation of PS1 by phospholipid flip-flop, the movement of phospholipids across the outer to the across the outer and inner leaflet in the membrane. And I would move on to the main topic, the role of PS1 in muscle-resident stem cells called muscle satellite cells. So skeletal muscle is composed of so many myofibers. Satellite cells, muscle-resident stem cells are activated upon muscle injuries. After activation of satellite cells are differentiated into myoblasts, and then myoblasts are fused to each other to become multinucleated cells called myotubes. Finally, myotubes are maturated to become functional myofibers. So myoblast fusion and subsequent myotube formation is crucial for regeneration of myofibers. So given that the membrane structure should be dramatically changed during myogenesis, we are interested in the distribution of phospholipids in the membrane. So it has been shown that the distribution of phospholipids are asymmetrically distributed. For example, phosphatidylcholine and sphingomyelin are distributed to the outer leaflet, whereas other phospholipids such as phosphatidylserine and phosphoinositides are exclusively localized to the inner leaflets. So it has been shown that the composition and localization of phospholipids are essential for a variety of cellular processes, including morphogenesis, apoptosis, and signal transduction. During myotube formation, previous literatures have shown that phosphatidylserine are exposed to the outer leaflet. Moreover, phosphatidylserine receptor called BAI1 can recognize the exposed phosphatidylserine, thereby promoting myotube formation. So we hypothesized that regulation of asymmetric distribution of phosphatidylserine may play a role in myogenesis. So far, the three types of phospholipid translocases have been identified. For example, phospholipid flippase are known to translocate phospholipids from the outer to the inner leaflets of the plasma membrane. So the transmembrane distribution of phospholipids should be regulated by phospholipid flippase, propases, and scramblases. But it is still unclear why distribution of phospholipids is so tightly controlled in the plasma membrane. To elucidate the mechanisms underlying myogenesis, we are also interested in calcium-permeable ion channels activated by membrane tension. This is because membrane tension is essential for promoting myoblast fusion, and mechanosensitive ion channels are thought to be involved in membrane fusion. But it is still unclear whether the phospholipids movement is involved in regulation of myogenesis. So we hypothesized that phospholipid flippase might be involved in regulation of PS1, one of the main mechanosensitive ion channels expressed in myoblasts. To examine the hypothesis, so we performed calcium measurements in phospholipid free-pace deficient myoblasts. Phospholipid free-pace is composed of the main unit P4-ATP acids such as ATP11A and so on, and is composed of the subunit CDC50. And we found that there are several P4-ATP acids are expressed in myoblasts, while only one CDC50, CDC50A, is expressed in myoblasts. And we performed calcium measurements on PS1 deficient, CDC50A deficient, and P4-ATPase deficient myoblasts. Using chemical PS1 activator YODA1, we found that strong calcium influx was detected in the presence of YODA1 in wild-type cells. And, but the calcium increase was clearly blunted in PS1 deficient cells. Surprisingly, we found that the YODA1 dependent calcium influx was almost completely blunted in CDC50A deficient cells and significantly decreased in ATP11 deficient cells. So this we thought that this is because CDC50A is only one CDC50 family expressed in myoblasts. So CDC50A deficiency caused almost complete absence of phospholipid free-pace. So we also confirmed the same phenomenon using human primary myoblasts, suggesting that phospholipid free-pace is essential for activation of PS1 in myoblasts. To identify phospholipid molecules that are responsive to PS1 regulation, we performed electrophysiological measurements using lysophosphatase serine. So lysophospholipids are known to be easily inserted into the outer leaflet of the plasma membrane. So we utilized PS1 expressing hex cells for electrophysiological measurements. When mechanical indentation was applied to HEC293 cells, we could appreciate the strong inward currents in control cells. However, the PS1 current was clearly blunted, clearly affected by application of lysophosphatase serine. Furthermore, removal of lysophosphatase serine from the outer leaflet by BSL wash clearly rescued the PS1 current, suggesting that PS1 on the outer leaflet may inhibit PS1. Based on our data, so we proposed the mechanisms called flip-flop switch. So during myoblast fusion, exposed phosphatase serine translocated to the inner leaflet by phospholipid free-pace. After the translocation, PS1 should form the active form that can sense membrane tension, thereby causing calcium influx that is essential for cytoskeletal reorganization, that is essential for morphogenesis of myotubes. So we proposed that flip-flop switch via phospholipid free-pace is essential for activation of PS1, thereby providing regulated myotube morphology. So let me move on to the main topic, the role of PS1 in muscle-resident stem cells called muscle satellite cells. So this is the cartoon showing the satellite cell-dependent regeneration of myofibers. Upon damages to myofibers, quiescent satellite cells are activated to differentiated and proliferated to repair myofibers by regeneration of myofibers or repair of injured myofibers. So during this process, muscle satellite cells are activated in response to various stresses, but it is still unclear how muscle satellite cells sense mechanical stress during myofiber damages. So we hypothesized that piezo-1 might be involved in sensing the environmental changes during myogenesis. So we checked the expression of piezo-1 in satellite cells. The in silico analysis showed that piezo-1 is clearly expressed in quiescent satellite cells. We also confirmed the expression of piezo-1 at the protein level. We utilized a mouse model called piezo-1-TD tomato, which can express endogenous piezo-1 fused with TD tomato at the C-terminus. So we can appreciate the expression of TD tomato in PAC7 positive satellite cells. So further confirming the piezo-1 expression in muscle satellite cells. To evaluate the piezo-1 function in muscle satellite cells, we established a piezo-1 deficient knockout mice. Given that systemic deletion of piezo-1 causes embryonic lethality, we established conditional knockout using PAC7 CRE-ERT2, which can express CRE recombinase in muscle satellite cells under the control of tamoxifen. Using the mouse model, we confirmed the deletion efficiency of piezo-1 in after application of tamoxifen by doing calcium imaging and checking the messenger RNA expression. To evaluate the piezo-1 function in satellite cells, we performed calcium measurements using calcium indicator PURA2. After isolation of muscle satellite cells from skeletal muscle, we performed calcium measurements. In wild type, calcium fluctuations were clearly observed like this, while the amplitude and the number of calcium fluctuations were significantly reduced by piezo-1 deficiency, suggesting that piezo-1 should work in satellite cells. To evaluate the function of piezo-1 during myogenesis, we utilized experimental model using cardiotoxin, myolytic toxin. After injection with cardiotoxin, we could appreciate complete degeneration of myofibers 3 days post-injury. However, regeneration of myofibers were observed 7 days or 14 days post-injuries. So using this system, we evaluated the piezo-1 function during myogenesis. So we first examined the muscle weight after myofiber regeneration. And we found that the reduction in muscle weight was observed after cardiotoxin injection in PD-1 deficient mice, implying that muscle regeneration could be delayed by PD-1 deficiency. By doing histological analysis, we found that several abnormalities, histological abnormalities, including neutrophil infiltration and collagen accumulation, were observed by PS1 deficiency, suggesting that PS1 is involved in skeletal muscle regeneration. To investigate why PS1 deficiency caused delay of myofiber regeneration, we examined the proliferation capacity of muscle satellite cells. After isolation of myofibers, isolated myofibers were cultured in specific medium. We examined the proliferation capacity by detecting PAC7 expression, the muscle satellite cell specific transcription factors. In wild type, the proliferation of muscle satellite cells was clearly detected like this, while the delay of proliferation was observed in PS1 deficient mice, suggesting that PS1 plays a role in proliferation of satellite cells. We hypothesized that PS1 could be involved in proliferation process of muscle satellite cells. To examine the possibility, we utilized PS1 TD tomato. After isolation of muscle satellite cells, we can appreciate that PS1 TD tomato was accumulated to the cleavage furrow during cell division. And we could see the immunofluorescent staining of PS1 TD tomato at the cleavage furrow during cell division. So we hypothesized that PS1 might be involved in progression of cell division. So to test the hypothesis, we performed time-lapse imaging and found that delayed cell division was observed in PS1 deficient cells like this. And interestingly, abnormalities in cell division, such as existence of chromosomal bridges and existence of binucleate cells, were observed in PS1 deficient mice, suggesting that PS1 is involved in progression of proper cell division. So recently, recent paper has revealed that the stiffness of muscle satellite cells is crucial for the functions under the control of a small GTPase called RhoA. So we hypothesized that RhoA signaling could be correlated with PS1. We examined the status of RhoA using rotekin that can detect an active form of RhoA. We found that the active form of RhoA was clearly decreased in PS1 CKO. Inconsistent with this data, we can appreciate that the amount of phosphorylated form of myosin light chain was clearly decreased in PS1 CKO. Moreover, consistent with the data, immunofluorescent analysis revealed that phase 1 was co-localized with low A at the mid-body of isolated satellite cells. Finally, we examined if pharmacological activation of low A could rescue the phenotypes of phase 1 deficiency. And we found that pharmacological activation using CN03 at least partially rescued the proliferation of mass satellite cells in phase 1 CKO. Moreover, low A activator CN03 at least partially rescued the abnormality in cell division, like this. So I'd like to summarize my talk by utilizing genetic mouse models. We found that phase 1 is involved in promoting myofiber regeneration and may play a role in a variety of muscle satellite cell functions, such as proper cell division. And as a mechanistic finding, we found that low A is one of the downstream effectors of PL1 in muscle satellite cells. So I'd like to thank the members of my laboratory at the University of Shizuoka, and we could see the Mount Fuji from the University of Shizuoka. And I would like to thank collaborators, and I would be happy. Thank you very much for your attention, and I would be happy to take any questions you might have.
Thank you very much. Are there any questions?
Very interesting story. I have a question. From what you were showing, I would predict that piezo is a fantastic indicator of apoptosis, because the appearance of phosphatidylserine on the surface of the membrane is one of the classic sort of signs of detecting apoptosis. And the next question is something in your developmental scheme maybe related to apoptosis?
Actually, so of course, we are interested in the correlation between PS1 and apoptosis, but we haven't checked the possibility. So I'd like to test the possibilities. And could you please tell me the second question once again?
No, was the question if your developmental scheme is maybe somehow related, if there is some apoptosis happening, which is sort of in the global picture of the development?
Actually, so we have tested the possibility of PS1 during developmental stages in skeletal muscle, and using PAC7 CRE that can express during developmental stages, we found that no pups were born in PS1 deficient mice, suggesting that PS1 might be involved in muscle development.
Very interesting studies. So actually, I have one question about the PAs. So it showed that actually quite striking that in the CDC 58 deficient cells, actually you could not detect any UDA1 responses, right?
Right.
So I'm wondering whether you have checked the poken response in that knockout cells, whether they can still respond to mechanical stimulation.
Yeah, that is a very important question. Actually, it was very difficult for us to perform electrophysiological measurements using myoblasts.
Yeah, actually, a lot of question is because you showed that the PAC can inhibit the channel from the out-leaflet. So I'm wondering what the kinetics of the flippers to kind of to exposure the PAs from intracellular side to the extracellular side, because piezo is really had a fast kinetics for activation. I'm wondering whether the kinetics could match.
Yes, so actually, so the kinetics of phospholipid flipping is quite high, quite fast. Probably, I forgot the exact number of numbers, but at least 10,000 molecules should be translocated from the outer to the inner leaflet per second. So the kinetics is very fast.
Okay, thank you. Yeah.
I have a question related to the cell division phenomenon. So you showed that piezo accumulates in this sect before division, and I was wondering if this is, does it mean that is migrating and concentrating there? Let's say that piezo before is diffused on the membrane and then it arrives in this position, and if this is physically driven by the curvature of the membrane.
Yes, so that is very important question. So actually, we don't know why piezo1 is accumulated to the cleavage floor. So as you suggested, membrane structures, curvatures might trigger the movement. And the second possibility is binding proteins might promote the translocation to the cleavage floor. So
that is on the plasma membrane or inside the cell? I'm interested in translocation of the piezo1 protein.
we haven't examined the possibilities, but yes, it is possible that piezo1 containing vesicles might be accumulated to the sites. Yes, but we haven't checked the possibility.
Okay, sorry. Any other questions? Okay, thank you very much.